Let's get started. Good afternoon and welcome to this session on non-viral delivery. So over the past year, we've seen the FDA approve some AAV gene therapies that deliver transgenes to the liver or muscle. And as we discussed in an earlier panel at this meeting, next-gen AAVs are making strides in maximizing the viral delivery capabilities in the CNS and elsewhere. However, for some applications, transient delivery or repeat dosing is needed, and lipid nanoparticles have emerged as a powerful technology enabling mRNA vaccines or delivery of mRNAs, genome editing components, et cetera, to the liver. Over the past few years, there's been considerable interest in extending these technologies to further optimize delivery to therapeutically relevant cell types, especially outside the liver. So in this panel, we'll be talking about innovation around delivery and cargo that can push genetic medicines into new applications and new directions. So I'm joined by our panelists today. We have Dr. Tom Barnes, CEO of Orna Therapeutics, Dr. Lou Brenner, CEO of Hopewell Therapeutics, and Dr. Guillaume Pepper, CEO of Senda Biosciences. So thank you all for joining us today. And to begin with, I'd like to ask each of our panelists to first briefly introduce themselves and their company's technologies, and also talk about what challenges in the current space they're trying to address. And we can start with you, Guillaume.
Absolutely. So very nice to be here, and thanks for the invitation. I'm representing Senda Biosciences. We're approaching deployments through a natural programming language we found through some work 4 years ago. We started to look at how species have evolved to communicate intercellularly with each other. And from that, we have derived what we believe a programming language. And we're trying to bring the programmable deployments comprehensively to programming nucleic acid and intending to program comprehensively the entire medicine, not just deployment on the left and nucleic acid on the right. And I'm very excited to be sharing what we've been up to with regard to addressing some of the challenges of a state-of-the-art technology.
Mm-hmm.
Great, thanks. So definitely glad to be here. I'm Lou Brenner, CEO of Hopewell Therapeutics. We are a venture-backed company focusing on novel LNP platform technology associated with unique lipid chemistry using a 4-component ionizable cationic lipid LNP that has been able to show delivery to a variety of tissues, including, not surprisingly, the liver, but at a higher level than has been seen with currently available lipids, and importantly, to a set of novel organs and tissues and cells, especially the lung, the spleen, and the CNS. We've been able to take our chemistry and pair it with biology to be able to show cellular level targeting. Ultimately, we believe for mRNA-based therapeutics or gene editing to get down to really cellular specificity, matching target and cell, and then also cargo design, being able to get increasingly complicated, large, and mixed cargoes for gene editing or gene writing to cells of interest. The challenge is obviously advancing that technology and financing the company in a pretty difficult environment for small companies.
Mm-hmm. Great. And Tom?
Thanks, Glenn. Also great to be on this panel. It's a very interesting topic. So I'm CEO of Orna Therapeutics. So Orna is a company that is creating products that are RNA encapsulated inside LNPs. So as far as that is concerned, it's, I think, fairly typical. The difference is, of course, our RNA is circular. And although circular RNA is known, and it occurs in all our cells, circRNA, that's how it's called in the field, ours was really the first engineered synthetic circular RNA that was made in vitro, designed to express and potentially be functional and have therapeutic applications. So we called our version oRNA, where the little o just simply means circular. Of course, so the company's Orna, but the RNA is oRNA. So we, in addition to that, we combine it with an example of extrahepatic delivery. We've in-licensed more than one and developed more than one delivery solution. But the one that we mostly talk about is our extrahepatic delivery, which is targeted to immune cells. And so the combination of those two is the premise that we use as our approach to in vivo CAR that we're doing. What we have, though, is a fairly broad-based platform. We struck a deal last year also with Merck, a fairly large deal, in the infectious disease area and also with a cancer application, not in the in vivo CAR specifically, but in a few other areas. So we feel very good that our platform, our approach is validated. And maybe the one thing to say about circles, certainly having worked with linear RNA in my previous company, which was in Intellia Therapeutics, that circles, I think, may emerge as the correct answer for what is the right format for biotechnology application for long coding RNA. I think if the world could do it all over again and everyone had access to the technology, I don't think anyone would use mRNA. I think it would all be circles because of the very large number of advantages without any disadvantages that circles have.
Great. So maybe building off on what Tom was saying in terms of the applications of non-viral technologies for targeting immune cells and that area, can you expand on what are the advantages and what's most exciting in this space in terms of translatability and application that can take the field beyond what we've seen with pre-existing technologies, whether that's linear RNA or viral vectors and the like. Maybe Lou, you can jump in there.
Yeah, so I think part of what Tom was saying is that the cellular targeting. So to be able to get not just the cargo, but to target the particular cell where you want that cargo to go. In the case of vaccines, dendritic cells and macrophages, for instance. In the case of CAR T, T cells. But I think that the expansion of the LNP field is being able to show fit for purpose. We're able to match the target cell, the lipid that will take something to that target cell, and also the package or the cargo that you need to influence that cell. Either to translation of RNA in some form, or gene editing or gene writing in that cell as well. And I think that's where the next generation will go. I think we've all benefited from the liver-directed LNPs for vaccines, for instance, but I think we see that as the tip of the iceberg for what could you do with this technology.
Yeah,
I think we're going to be probably in the same line of precision medicine at the end of the day. Nucleic acids are great to operate within the cells, but they need the zip code to get to the right cell, and that's what's missing. And I'm really thinking about the comprehensive program of medicine that we started to touch on. With this technology that is emerging, we have for the first time the ability to be very comprehensive in the way to design medicine, and you do that in a programmable way. For the vaccines, I would slightly disagree that liver is not the place to go to create an immune response. And the current commercial products, at least for the European submission of one of the two commercial products, indicate less than 1% in the spleen, which is the proxy for lymph node targeting. Most of it is muscle and liver, but has probably not much to do with an immune response. So precision, bringing the mRNA to the right place at tissue level, lymph node, and then at cellular level in the germinal center, get you going with an immune response that will be much better, broader, maybe multi-strain, also mucosal and also systemic immunization, which is kind of the holy grail for any vaccines. And then you can go into T cells. You can start thinking about in vivo CAR if we were able to deliver a CAR mRNA to the right cells, T cells, and express on the surface. With that precision, you will have likely a affordable in vivo CAR, and many patients will benefit from this technology. So precision is a key word that comes to my mind with your question.
Got it. So I guess how granular can we get on this front? There's always going to be different subtypes of T cells, of monocytes, of things like that. How should we think about how granular the therapeutic space needs to be, and what do we know about how this works from a mechanistic perspective?
Maybe I'll take that. To me, delivery is very much also the art of the possible. If you could deliver to a particular site, you could write down the list of things you could do with a particular payload. And obviously some sites, there are one or two diseases or maybe one big disease, and then other sites, there's a whole slew of things. I think one of the reasons why there's interest in delivering to immune cells on the fly is because there are actually a whole bunch of stuff you could do there, some of which addresses quite large populations and I wouldn't say an unmet need, but certainly in a much more improved experience both for the patient as well as for payers and so on, the cost of some of these therapies, whilst striving to sustain the response seen. I think it's some of the, I think why in vivo CAR is so interesting is because the performance is there with cell-based therapies, and is there a way to just make all the fuss and bother of that go away and make a much simpler approach? So it kind of lends itself to that in terms of a pull, but the question is also the push. Just because you want a delivery that goes there doesn't mean you're going to get a delivery that goes there. And so people have tried to drive particles to particular sites by combining with other technologies, which has worked to some limited degree. We favor, I suspect Lou favors also as well, the classic 4-lipid LNP. It's the one thing really shown to be scalable. I certainly hope for Guillaume that his particles also prove to be scalable, because it looks to be quite interesting. But being able to first figure out where you can go and then decide the things you can treat. So there's both what is enabled by the technology at hand and also on the other side where you'd want to be, and one is trying to sort of put those things together.
Maybe one word I want to add, creating a bit of a reflection on the comprehensive part of it. You could achieve targeting through the payload and the delivery, right? So the comprehensive nature of what we're trying to attend could be amplified by looking at the full medicine, not just a part right here, which is that zip code with a nanoparticle, and totally separately the cargo. By embracing comprehensive programming, if that becomes technology possible, you can amplify the ability to be precise.
If I could add, the complexity of the cargo is, I think, one of the next that you referenced, that the field has really, I think, scratched the surface of the interface of the delivery and the cargo. And the complexity of the cargo, the size of the cargo, the nature of combined mixed cargo, the gene editing apparatus, mRNA or RNA and DNA together, and then importantly on top of that, the ability to redose. These are the things that I think are really unmet needs for the field, and I think that each of these companies is trying to work on.
Yes. So how far behind are we in terms of putting in cargo aside from linear mRNA? Obviously, this is something Orna is thinking about very carefully. But so where is the field in terms of bringing things beyond linear mRNA in lipid nanoparticles?
Maybe I can. You want to go
Go ahead.
I mean, we are approaching the deployment through a very unique technology, so a lot still need to be de-risked and advanced, but being part of the flagship ecosystem, we had played with a lot of different next generation RNA, and have been able to accommodate DNA, a bunch of different RNA, including the mRNA that we build a factory for at Senda. We can deploy protein peptide, we can multiplex with we have a study with mRNA CRISPR-Cas9 and the guide RNA, and all of that could be packed so far into our technology. So there will be limitation, but we haven't necessarily just yet hit the ceiling.
Yeah. Interesting.
Yeah,
One of the challenges, I think, is also very practical in the lipid components and the solvents needed for them are generally not compatible with proteins or proteins maintaining their shape. So that's sort of the, let's say, the first barrier to just putting a protein inside a classic LNP. Now, I'm sure creative people could turn their attention to that, and certainly if you're not using a classic LNP, there might be ways around that. But the application space for solving that is, I think, quite huge when you think about editing, which is currently done in the LNP space with a long coding RNA and a short guide RNA, which is chemically, not biochemically made. It would be much more straightforward to package the pre-assembled editor RNA complex. Now, if you could do that and then put that in the particle, then that would be a plus.
Yeah. The other aspect is, the mixed cargo or the DNA. If you really want to get to, true gene editing, I think there's a pretty consistent call for the ability to deliver DNA. And we've been able to show in some pilot experiments the ability to do that. Again, matching the particular cargo, the length of the cargo, and the particular lipid, that's really where the art is.
And one of the things is also that we tend, or you often encounter people who think that there's an aqueous, a watery environment inside the particle, which is not true for classic LNP. All of the molecules are pressing up against each other, so it is hard to package stably a very charged molecule like a nucleic acid and a much less charged molecule like a protein all in the one, because your tendency is going to be for those areas to separate out and not be a and that's going to be a problem. So maybe Senda's particles have a different approach there. I don't know if they are aqueous in the middle or whether you believe they are, Guillaume?
No, actually.
Yeah, no, but it sounds like there's a very real biochemistry specific considerations for the type of cargo that can be delivered that need to be engineered or figured out for the different platforms, and that's what's going to be shaping how the different technologies advance for these different buckets of applications there.
Yeah.
So maybe coming back to the tissue distribution, what are the other cell types that maybe we're not thinking about yet with respect to non-viral delivery, but that could emerge as being exciting?
So maybe I'll talk a little bit about the lung, because I think that's where we've most developed our own story. I think the unmet need is to get down to the granular. Once you get beyond the tissue, you think about, okay, what are the cell types within the lung? Whether the endothelial cells that open up possibilities for diseases like lung cancer or pulmonary fibrosis, or pulmonary hypertension. When you think about the epithelial cells, diseases like cystic fibrosis, and you get specifically down to the basal cell. And then when you think about inflammatory cells or macrophages, you think about diseases like pulmonary alveolar proteinosis, for instance. And so I showed in our presentation RNA-seq data that's quite elegantly published in the last year that really matches the cell type with the gene transcriptome. And that almost gives a treasure map for where you would like to design your LNP and your cargo together to be able to hit those cells. And I think that exists in other tissues as well, but that kind of level of specificity, just getting to the organ is, I think, the first step in the process, but getting down to the cell type and then knowing the gene, that is all where I think things will eventually move in this precision sort of strategy.
Guillaume, what about you guys? Senda has obviously done some work in
we've done a bit of work on the lung, epithelial cells. Maybe just to kind of depart a little bit for what I've seen so far, we are at the start of looking at oral for nucleic acid.
Yep.
And we can turn this particle to target pair patches, for instance, within the lining of the small intestine or epithelial cells, one or the other. We can turn this particle to traffic through the lymphatic system to pancreas through an oral gavage. It's mice work for now. And we also can turn this particle to express the mRNA expressing the protein that get in the circulation. So there's a lot of tunability here that we started to learn from what I believe nature provided to us. And hopefully if this is a natural programming language, that's we may all of us looked forward to program the deployments.
Yeah. So maybe coming back to the cargo components, I think some of you alluded to also engineering the cargo and how that could be co-developed in combination with engineering the nanoparticle or non-viral component. Can you expand more on why that's important and how that can help increase the activity or the specificity of what you're delivering, either for vaccine or gene replacement or CAR T or other applications?
I think actually we have a very interesting example, which I didn't have a chance to get into detail in our presentation earlier today. But the difference in shape of RNA and mRNA versus RNA changes the biodistribution of the underlying particle. And the way to think about that is that we think it's primarily driven by a tail. The body of an mRNA will adopt some sort of secondary structure as it tries to minimize its free energy, but the tail can't pair with anything. And so with multiple tails in one particle, they will tend to phase separate, and that will create misshapen particles, and misshapen particles tend to go to the liver. Circular RNA has no tail, and so it packages much more readily, much more consistently. And so it will reveal more the true tropism of the particle rather than sort of default, crummy particle pathway which goes to the liver. And so the nature, that's just an example of how the nature of the payload chemically, biophysically, influences the particle, which influences the protein corona, which influences the biodistribution.
Good. I'd take it a slightly different direction. I appreciate that answer. The other thing that I think is underappreciated on the outside is the nature of formulation and stability of these particles. And if you think about the COVID vaccine example, for instance, everyone remembers the -80 freezers and all the different sort of time associated with this. And I think one of the needs for the field is how do you make these therapeutics really commercially viable? Making them commercially viable, obviously you have to manufacture them, but they have to have some degree of stability. And a lot of effort within companies, including ours, goes into, okay, how do you ensure stability, repeatability, freeze thaw, and those kinds of things. And so I think there is a nature of not only the lipid itself, the combination, how do you do the microencapsulation, and then how do you formulate it in a way that's stable for as long as possible. And so I think that's a really important part of the field that may be underappreciated outside the corporate environment as well.
I can maybe add one word. You looked at a cell phone high-end product. It's actually old one, but very high-end product, designed integratively from the view of what need to be the product to the actual product. I think the future of medicine is to bring medicine to an high-end product and to the benefit of the patient. And the technology started to be available to have this precision and open the way for integrative design, which was not necessarily available to us. To these days, we can, again, program nucleic acid. The deployment is not programmable. If you start being able to program deployment, you have the missing part that allow maybe all of us to enter into a space where we can have integrated design capabilities and bring medicine to the high-end product you would expect for anything that you were, as a customer, experience with, like a cell phone.
Right. Yeah, so it sounds like there's multiple levels of engineerability, both in terms of the chemical composition of the lipid or other components of the nanoparticle itself, the formulation of what's inside, and then the biochemistry of the cargo, and also the sequence of the cargo and whatever you're encoding in there. Presumably that factors into the overall profile of the therapy as well.
The mistake will be to continue with this emerging technology to see the world in two parts, cargo, delivery, and ignore the fact that you can have this integral view of what the medicine need to be. And then you throw the stability, you throw a lot of things that today we second guess, and then we get too late and we realize it's not stable enough, or we carpet bomb mRNA away from the target, and you get vaccines that save the world, but a lot more can be done. Right?
And I think a part of it too is off-target effects. I think one of the most frequent questions that we get is, don't show me where it goes, but also show me where it doesn't go. And so there's a really important sort of nature in the field for biodistribution, both organ and cell, because at the end of the day, and I'm a kidney specialist, when I think about first-generation nucleic acid therapies, the renal indication was how much damage is it doing, as opposed to how much positive effect is it having in other places. So.
So to what extent are we, I think I've seen a number of presentations now on various non-viral approaches. A lot of the discussion is getting away from the liver, basically. To what extent is that doable with current technologies? Is there further optimization that is needed? Kind of how are we on kind of solving that issue?
I think there's a couple of approaches in play. The tropism of a classic LNP is determined by the protein corona that it attracts. And the proteins that it attracts are based on a low affinity, high avidity interaction between the lipid, typically the head group of the lipid, and stuff in the blood. Now, ApoE is a very available protein to pull onto the surface of a particle, and if you do that predominantly, you will be a hepatotropic lipid because the ApoE receptor is found in the liver. So the question is, what other proteins are even available for a classic LNP? I mean, obviously Senda's in a little bit different place, but for a classic LNP. Well, we don't have that catalog. So to some degree, it's out there, but you have to go discover it, and typically you have to discover it empirically, because even in vitro, you can't recapitulate the process.
Exactly.
And so it's very laborious, but however, when you do get one, it can be extremely valuable. So we feel very grateful, for example, at Orna, that we do have a very nice immunotropic lipid, and so we're of course going to make the most of that we can. Of course, if you go away from the LNP world, the rules for what governs biodistribution remain to be discovered. I don't know, Guillaume, if you have a good sense of it yet. I mean, it's phenomenology. It goes here,
Trying
goes
to
there.
understand how it works, to be very clear.
Yeah.
The other tissue that I'll sort of highlight where we have some initial data, and I think is one of the holy grails of the field, is the central nervous system. How can we get increasingly complicated cargo across the CNS done systemically? There's certainly been a variety of approaches, ICV or intrathecal, which are probably not scalable other than really extreme clinical situations. But we've been able to design initial LNPs using motifs from neurotransmitters with the mechanistic sort of probability or belief that those are picked up in some way, maybe in the blood themselves, or maybe at the blood-brain barrier, and carried across to different cells of the CNS. And that's sort of one of the, I would say, from Professor Zhu, one of the clever innovations to be able to think about how do we go to different places, what are the biological mechanisms that you can take advantage of, protein corona, transmitter pathways, to be able to carry your cargo using biology to work in your favor.
It's interesting to see that after all of these years in VNN, eposome, LNP, we're still trying to understand how it works, and I'm glad you touched on protein corona. It's such a complex things to try to address. And the milieu may be different depending on route of administration and what you're trying to do, which add complexity. Disease patient versus non-disease patient may add their complexity to it. But I'm hopeful we can figure it out and open a new era for the industry.
Yeah, it's a good point. Even IM versus IV, that those have very different sort of drainage pathways, and where you go first pass, in addition to the liver being the protein corona for the ApoE dependent mechanism, it's also just the first flow of, most intravenous injection. And so be able to avoid that, to be carried to next tissues. The lung is next, then the spleen comes along, and so all of that is part of the pathway.
Many times I think people have successfully drawn from nature and say, well, how has this problem been attempted in nature, and what's the answer there? That's viruses. That's how viruses do it. But I think the reason why, and we've certainly used viruses, I think, as you started off with AAV, but I think one of the reasons why viruses have a problem is because viruses are solving for more than what we want. Viruses not only have to get somewhere, and of course with a protein coat you can evolve that to exactly the specificity you want, but it has to replicate and be packaged and get out of the system and retransmit. We don't need that. So, and we ideally need redosability. So there's aspects of the viral answer which isn't actually useful for what we're trying to do. Maybe the exosome concept, or other kinds of natural particles maybe that's a closer analog, but certainly the LNP is a completely sort of ab initio concept, has been evolved to a point where, by people, where it's kind of functional, but we don't have a close analog. Right. At least that's widely accepted, of something in nature that we just need to copy.
That may be where we're bringing something, at least from an inside. Let's see how it goes. But it is a fact that species have evolved to communicate intercellularly in any kind of milieu or ecosystem, certainly in our body as well. So the intent here is to mine the way this communication have been happening through millions of years of evolution, understand the chemical language that allow this directed communication, repeat and self-dosing, and pick up the language and then understand the rules of the game. So there is a lot of parallels with the genetic code, for instance, or maybe the attempt we had using AAV or virus as a kind of vectors for deploying molecules as well.
Another point we haven't touched on, I think it's important, is biodegradability. I think that it's very clear that many of these eventually make their way through the liver. But understanding the breakdown pathways of the LNP, of the lipid itself, and even the RNA usually you would presume would be broken down in the cell. But understanding the biodegradability of lipid, how long it lasts, where it goes, and part of the ionize of a cationic lipid, the design is so that can go through natural kind of degradation pathways as well.
So as we think about where the spaces, the lipids that are available now, the nanoparticles that are emerging, it sounds like a lot of it is so far still gleaned from empirical studies or screening, and you screen for something, you pull out what works, and then characterize and pick the ones that develop best. So how far away are we from being able to program things more directly? You did touch on some strategies that incorporate, let's say, a targeting domain or some kind of ligand onto a lipid nanoparticle. Are there drawbacks to that make that challenging? How can we make this more programmable?
Yeah, I think some of the tissues that have proved harder, the kidney, for instance, you may have to do some kind of ligand, or the muscle, maybe a ligand-based approach. Other tissues, it is a degree of empiricism, but there's also intention. Like I used the example of the neurotransmitter, putting those into the LNP with the belief or expectation those would go to the nervous system in some way. So I think it's a combination of both. We have a slide that we show like it's an iterative process where there's some degree of intention, some degree of observation, and then a bunch of repeating. And if you sort of carry it to the extreme, ultimately AI may be able to be used to sort of run that virtuous cycle more efficiently.
It's also, you want to think about what is your target. If we're talking quantitatively or numerically, what is your target? So if you're going for a genetic disease, and therefore you need to get it to a certain site of tissue, every cell is mutant, even though it's not expressing or doesn't require the function. So it certainly, I don't think matters that your stuff goes everywhere as long as it gets to where it needs to go. And if you could dose it safely, maybe the answer is just give more until you get enough at the place. So there's maybe more than one way to achieve what you're trying to achieve. It may not require enhancing your specificity. You could decide not to care about all the stuff as long as it's safe.
And for us, the platform is set up to work at more than 100 test articles per week, and we have started to put computational science to give us in 2 weeks 40 test articles a performance by design. So the approach is to basically set the performance, find out of 75,000 molecules that we have in FIS Atlas, far exceeding in molecular diversity what is available with synthetic lipid. So it's how do you pick up the right composition to achieve a given performance. So at the stage where we are 3 years in, it's a 2 weeks 40 particle. We're multiplexing right now, we're automatizing, and the goal is to your point, start maybe applying some machine learning capabilities to learn faster how this language work, and hopefully continue to progress the science toward an exciting new target.
So looking ahead, where do we think the space could be in 5 years?
Comprehensive programming of medicine.
So all of these challenges will have been achieved 5 years from now?
No, there
That sounds
will be
pretty good.
limitation. But I think Tom is putting the finger in the right place. When you start moving into, for instance, immune cells, you have a wealth of disease you can get into, and you have the precision, you have the selectivity. You can also bring different type of molecule to or combination of to enter and address patient needs. So you don't need to solve all of the tissues and cells of the body to start having a massive impact to humankind. So
yeah, pleiotropy can be your friend in the immune system. One of the things that we like to dream about, certainly within the next 5 years, is the idea that since we are in multiple lineages with one administration, we're in Ts, NKs, and MACs, it's not just about leveraging the function of those cells with the same circle, but designing a product which has multiple circles where they're designed to function in the individual lineages. So we're talking here not just about systems biology, which is already fairly high science, but is typically thought of within one cell. We're talking about systems biology across cell types, lineages, which is not something anybody's doing right now. But it doesn't require anything other than what we have today. It just you just have to sort of work the problem.
I think there'll be more specific lipid technology. If you look at the lipids that are out there now, those in Alnylam, Pfizer, Moderna, those were designed, initially tested and deployed decades ago, really. And so I think, but they've also paved the way for the next sort of generation. And I think that the newer lipid delivery-based mechanisms will carry things either better to the liver or to additional tissues that open up, different organs and different diseases to treat.
So do you think we might see proof of concept in some other organs beyond the liver?
Yeah.
For sure.
We're actively working on muscle, for example, because we can make the long RNAs for the long genetic muscle disease. But no, I think there are a number of really high-value tissues out there, and people are going to be plugging away at it.
Great. So in the last couple of minutes, I also wanted to touch on manufacturing. That's a big thing in the viral genetic medicine space. How do the current platforms that you're working with fit into what we think of as the manufacturing workflows for lipid nanoparticle or mRNA technologies?
So I'll take the first shot at it. So it's complicated in that there's multiple parts, right? It's the RNA, and there's certainly traditional ways to synthesize that, or the mixed cargos if those are coming together. Then there's the lipid itself, and then there's the combination or the encapsulation. So it's really a multi-step process. And then, as I referenced briefly before, the formulation, how those come together for stability. We have been able to scale, for instance, from mouse experiments to non-human primate experiments, but it's a lot bigger scaling to go to human experiments to do that. And so I think there's been a lot of build, and we interact with a lot of manufacturers. There's been a lot of build of capacity in the space. And I think that's paves the way for everyone. But really the devil is in the details. How do you get that encapsulation work done together? Do you make the lipids in one place, the nucleic acids in another, and then bring them together in a third place and fill finish in another? So I would say it's a long answer, but it's a complicated sort of multi-set of environments. The good news, I think, is the capacity is expanded, and tech transfer is possible now in a way that it wasn't possible a few years ago. If you think of what happened, again, with the COVID vaccines, how quickly those came together.
Everyone agrees?
Yeah, I mean, I think billions of doses of the vaccine has been made, so that shows that scale is possible, and that's been mostly done by 2 companies. So I think that as long as you're making a similar product, and I believe Hopewell and Orna is making classic 4-lipid LNP, so that should scale. We like the fact that the circles scale actually more readily than mRNA does, so I think we're in good shape there. But yeah, I think as you start doing anything that goes off the beaten path, functionalizing the surface of a classic LNP or going to a different style of particle, I know there's been a long-standing interest in the polymer nanoparticle, which we haven't talked about at all today, but that's always been something that's out there. You'll have to address those questions afresh.
And for Senda, since we're bringing something a bit different, we fit right into the nanoassembly used for LNP and RNA. Very sadly, one part which is new is the extraction from natural source and the fractionation that provide the components that can then fit the nanoassembly. So I don't anticipate necessarily a lot of complication from a manufacturing viewpoint. Even if I think we need to apply advanced analytics to understand carefully what's going on as an input, and then characterize the full product after we nanoassemble the LNPs of the mRNA and the deployment system.
Yeah, perhaps things can get a bit more complicated when the cargo becomes a little more complex as well.
That's for sure.
Yeah, there's a world of, we didn't touch on it, but a world of analytics. How do you prove what you've done on a consistent, repeatable, sort of stable basis as well?
Yeah. No, is that something that's an open question, or is that also something that can leverage what's been done with the COVID vaccine space?
Yeah, I think a lot of us have read the summary basis of approval for the COVID vaccines, because that gives at least the minimum requirements for what would be necessary. But obviously, the devil will be in the details for every product. But I think it paves the way for the field for sure.
Great. I think we're just about at the end of the session. So I'd like to thank our panelists again. That was a nice, lively discussion.
Good. Thank you.